And this is all just attributed to GMO 1.0.
Yes.
Now there's something of a 2.0.
And let's get into the heavy stuff.
Let's go into that because you've been preaching about this and awareness of the dangers of GMO 2.0. So tell us, what is, what's the difference between a 1.0 and a 2.0 GMO, and why should we be scared?
Well, and there's, just as there's a way out of GMO 1.0 by eating better and then convincing many people, so we have a critical number, which creates a tipping point of consumer rejection, which is actually underway. Now we have GMO 2.0, which are new techniques which have the ability to create GMOs very, very quickly, very easily, very inexpensively, and some with more power. So there's gene drives where you can create a genetically engineered organism and all of its offspring get the traits instead of just half from the mother and half from the father. There's sprays that can, with RNA in them, that can kill insects and possibly get absorbed into our system and reprogram our DNA. There's insects that are being developed, not yet created, with genetically engineered viruses where they can do genetic engineering in the field. There's all sorts of genetically engineered environments that they're envisioning, not just GMOs, but let's change the microbiome in the soil. Let's change the insects. Let's spray with RNAi and create GMOs. The most significant change right now comes from gene editing like CRISPR. Mm-hmm. Now, those are ways to create GMOs that are cheaper. And the way that they do it in the past was they would shoot a gene gun of millions of particles coded by genes, hoping that some of them make it into the DNA of some of the cells, or smuggle in genes in bacteria, but you don't know where in the genome it ends up. Here, like with CRISPR, you have a scissors and you have a guide. Okay, cut here, it cuts. Cut here, it cuts. Now, the biotech industry has been pretending, just as it did with GMO 1.0, that it's precise, safe, and natural. But they've convinced governments that it's precise, safe, and natural. So governments like the United States, Canada, the UK, India, Australia, Japan, Argentina, Brazil, have believed them and deregulated gene editing plants, animals, and or microbes, especially if there's no foreign DNA put in. But the technique is far from being ready for prime time. The journal Nature describes the outcome of 3 CRISPR experiments on human embryos as chromosomal mayhem. What happens is it can cut all over the place, not just in the place you want. When it reconnects, it can cause deletions, additions, actually chromothripsis, which is the shattering of the chromosome and the reorganization. It can pull DNA from other sources that happen to be in the petri dish or used in bacteria as a process of genetic engineering. It can, and we have a little 6-minute animated film called 7 Reasons Why Gene Editing is Dangerous and Unpredictable at responsibletechnology.org. That is a really important 6 minutes because it shows the elected officials and regulatory agencies that they've been lied to. And it names 7 reasons, so it becomes a challenge. Show us, biotech handlers, that you've not been lying to us about these 7, and they can't. They don't have science on their side. But they do have recruited ministers and ministries and secretaries of whatever claiming that, oh no, it's safe and easy. So you can gene edit a food in the United States and put it on the market without telling anyone in the government. And if you send a note to the USDA, they'll say, oh yeah, it doesn't fall into our criteria if it doesn't have an inserted gene from a known pathogen. So go ahead, put it on the market. And Conscious Foods, which is a terrible name, but they're gene editing salad fixings and putting it on the market unlabeled because they're doing it through restaurants and caterers. So you may be eating GMO stuff that no one is able to identify. Now, in the short term, these stealth GMOs, which could create new allergens or toxins, could flood our food supply and cause disease. That is a catastrophe, but it's the least of the worries. On a bigger scale, there's a gene rush because it's so easy and cheap to use CRISPR. You can buy a lab for less than $2,000. All these people want to use gene editing to solve a problem or make a fortune, and sometimes both. And they're targeting insects and trees and grass and birds and pets and livestock and all sorts of types of food in hopes that their patented gene-edited GMO could solve a problem and make them rich. Now, when you release them into the environment, they can't be recalled. They self-propagate. The most common result of genetic engineering is surprise side effects. So now you have an unregulated, easily accessed technology that is prone to side effects, that is permanent in the environment, and that is the recipe for disaster, where we will incrementally and then wholesale replace nature in as little as one generation. So future generations will not inherit the products of billions of years of evolution, but a mix of natural and man-made prone to side effects that you can't even tell. So this replacement of nature is the fulfillment, I know, of an indigenous prophecy made by one wise man who told a friend of mine in the early 90s, we have the capacity around this time to end biological evolution as we know it, unless we do something to stop it. I heard those words and go, that's the best definition of what's happening I've ever heard. I think he was talking to us.
Yeah.
So that's a bigger piece where we have come to the inevitable time in human civilization where we can redirect the streams of evolution for all time, on purpose or by accident. So now is the time to make important decisions about protecting all living beings and all future generations, rather than haphazardly being the bane of existence for future generations, dealing with the folly of our lack of ability to step up and own the fact that this technology is so powerful and long-lasting. What we've done at the Institute for Responsible Technology, we're joining the global campaign to try and get the regulators to re-regulate, or to, in the EU, to stop the deregulation. But we're focused on the most dangerous GMOs. The most dangerous are the microbes.
Mhm.
Now, we know from COVID, whether it was genetically engineered in a lab or not, it could have been. And there's an example of a GMO potential to take out millions of people, far more than this pandemic, because there are far greater potential pathogens that could be enhanced through genetic engineering. But infectious disease is one small sliver of what a genetically engineered microbiome could do. So all those who are trying to stop the gain of function research on potentially pandemic pathogens, good on you, but not enough. You're missing biology. You're forgetting something. Over 50,000 studies in the last 5 or 6 years have shown that the human microbiome is fundamental to human health. We outsource about up to 90% of our metabolic and chemical functions on a daily basis to microbes. Things we can't do on our own. We co-evolved with the microbiome. And so, okay, you do this better, you handle that, we'll handle this. And a huge amount is outsourced. In fact, we can get away with a measly 23,000 genes, less than earthworms, because we use the 3.5 million genes of the microbes living inside us. And they do things that we couldn't even design with all of the best scientists, let alone with our bodies. Now, it's so important that the human microbiome is designed and healthy from the start, because the entire health of the person's life can be dictated by the population and diversity of the microbiome in the first few months. So milk digesting microbes enter the birth canal in the second trimester. The baby is born and inoculated. The milk, the first organisms that come from the breast, clear out the oxygen from the digestive system. So it's an anaerobic, non-oxygenated area for the best microbes like bifidobacteria. Then 27% of the microbes in the body of the infant come from the milk, 10% from the skin on the nipple. And if the baby is feverish or has a problem, their salivary microbes communicate back through the breast, changes the milk supply, and changes the health of the body. And a large portion of the milk is undigestible by the infant, not designed for the infant, it's designed for the microbes. Why? This This incredible system is designed because microbial integrity is so important. Now, what's interesting is this. Microbes not only travel and mutate like we know from the pandemic, but they swap genes with other microbes. That's why they're so intelligent. They swarm their genes so that they can better support ecosystems and organisms like humans and be supported by. So you release a genetically engineered microbe, and let's say it travels around the planet, which it can. It mutates, which it can. But the altered gene that you created and released in soil now ends up in the gut of an infant on the other side of the planet. And it's not even the microbe that you created. It's something else. It's been changed. So you have a nitrogen-fixing microbe like they're having 3 million corn acres right now that's genetically engineered. What happens if you have nitrogen-fixing gut bacteria? We don't know. Similarly, it'll end up in other ecosystems, doing things that were never intended or anticipated. And again, the most common result of genetic engineering is surprise side effects. But with microbes, even if it happens to be precise and with no side effects and no mistakes, you're changing the microbial nature of organisms that are fundamental to human health and turn out to be fundamental to our planet. Algae creates most of the Earth's oxygen. Fungal networks shuttle nutrients between trees. Soil microbes not only create healthy crops, but sequester carbon. All of this is in danger if we put out genetically engineered microbes. There's an example of one that was created in the early 90s. Well-meaning scientists, let's solve many problems at once by genetically engineering a bacterium that survives in the root area of every plant on Earth. This is from a film I created called Don't Let the Gene Out of the Bottle. 16 minutes, watch it at responsibletechnology.org. They took Klebsiella planticola bacteria and turned it into a still to create alcohol. You can feed it plant matter and it turns it into alcohol. So they said, great, let's feed it all the crop residues that are normally burnt, put it in a tank, put the bacteria in, create alcohol for the tractor of the farmers. They can sell it as fuel, they can drink it, and they can take the nutrient-rich sludge at the bottom and spread it on the field as fertilizer. Well, you'll see in the film that there was a graduate student that happened to be doing research on this genetically engineered microbe that had already passed all of the EPA-required studies and was about to be released into the environment, and discovered that the nutrient-rich sludge, if it had been spread on the fields, could have theoretically rendered those fields sterile, because it could have turned all of the plants into alcohol, starting with the roots. And we also know from whistleblowers talked about in the film that a secret EPA study found that genetically engineered microbes go around the Earth. You can find them everywhere. So I asked Dr. Elaine Ingham, who's now like the queen of soil biology. At the time, she happened to be the advisor for this graduate student wanting to get his PhD. I said, what would have happened if they released it? Worst case scenario? She said, an end of terrestrial plant life on the planet. That's a cataclysm. But that cataclysm, CASPAR, is a microbe doing what it does. It's designed to do, genetically engineered wise, but in more ecosystems than planned. That doesn't even consider the mutation or what's called horizontal gene transfer, where that alcohol-producing gene now ends up in guts, making us drunk, in oceans, doing whatever it could do there. It doesn't take into account what could really go wrong, and already it could produce a cataclysm. There's another microbe in the film that can potentially change weather patterns.